And I will just concentrate today on the introduction to the respiratory system. You try to have a notebook with you right now, and then whatever I am saying, whatever things which I want you to note down, you please note down. Are you having the textbooks? I don't know how many of you are having the textbooks also. Let us proceed with the topics. So the word respiration has been derived from the Latin word respirate, means to breathe. You have the human respiratory system. Are you able to, I think you are able to see this picture. You have the upper respiratory tract, you have the nasal passage, and then this is covering the picture. This is the cover. You can just remove this portion. You have the nasal passage, and then the oral cavity, the pharynx, the larynx, and then you have the trachea here, and then the bronchi, and then you have the lungs and all that. And in addition, you also find the heart and other things. So all these structures constitute the respiratory system, the anatomical structures of the respiratory system. And these slides are not moving. Fine. So why do you require respiration? Actually, you have the carbohydrates, and these carbohydrates require oxygen for oxidation, and that in turn will split into carbon dioxide and water molecules, and then ultimately release energy. Energy is required for various metabolic process, and for everything you require energy. Energy is being released by the combination of oxygen molecules with sugar molecules. And then you have to understand that there are 300 million alveoli in each lung. And the total surface area of both the lungs put together is 90 square meters area. All the alveoli, everything put together is 90 square centimeters. And the diameter of each alveoli is very small. It's about 0.2 millimeters. So the ultimate fundamental unit of the respiratory system, the alveoli, is 0.2 millimeters. And there is what is called the alveolar capillary membrane, the term. Note down the thing in the notebook, alveolar capillary membrane. Alveolar capillary membrane is between the alveoli, that is, as the air that comes in contact with the respiratory membrane, that is the fundamental unit. And just adjacent to that, you have the capillary. So the blood flows there. And this particular thing is called alveolar capillary membrane, wherein the oxygen molecules diffuse through the alveolar capillary membrane and then tag on to the hemoglobin molecules present in the RBCs. And these RBCs are present in the capillaries. So the width of this area is 0.005 millimeters or 0.5 microns in distance. And during quiet breathing, 250 ml of oxygen is normally being utilized in the body per minute. 250 ml of oxygen. You will be using 250 ml as you are sitting in your place and seeing these PowerPoints. And let us say you are subjected to exercise, or you are running to catch a bus or to catch your class as the time is getting on. And then it may be increased to 20 times. That is nearly about 5,000 ml in exercise, in severe exercise. And then you must, one more thing I just wanted to tell you at this point of time is try to practice diagrams. And the diagrams goes a long way in understanding the subject. At the same time, you can readily recapitulate the details of the subject. The right lung has 3 lobes. So practice the right lung and then divide them into 3 lobes. And then you will be having one oblique fissure and then one horizontal fissure that makes them into 3 lobes. And the right lung has 10 bronchopulmonary segments. This point you must remember. Right lung, 3 lobes, 10 bronchopulmonary segments. These are the commonly asked questions in the Viva. Or remember, all these things which I am mentioning, you please note down in your notebook and then prepare notes for this. And the left lung, the right lung, 3 lobes. Left lung has 2 lobes and has 8 bronchopulmonary segments. So this is the broad anatomical distribution of the lobes in both the lungs. And you must remember, just try to breathe in and breathe out. Inspiration is for a period of 3 to 5 seconds. Inspiration is for a duration of 3 to 5 seconds, whereas expiration is only for 1 second. So you inspire longer, and expiration is only for 1 second. This point you must understand. And this knowledge of inspiration and expiration will be useful when you are doing the practical classes about the respiratory system. And just like any other intensive units, for example, intensive cardiac care unit and intensive pediatric care units, you also have an intensive unit in the respiratory system. So the diseases which occur in the respiratory system for which you will be giving intensive care is the respiratory intensive care unit. So the respiratory intensive care unit where the person is not able to breathe or there is any damage to the lungs or because of any other cerebral or other conditions, this will be changing. And the knowledge of pulmonary physiology will be useful in branches like anesthesia and medicine, pediatrics, cardiothoracic surgery, and pulmonary medicine. So all these people, all these branches and all these specialists will be available in intensive respiratory intensive care for the Medical Council and National Medical Council. So there will be stipulated 5 beds available in each hospital for respiratory intensive care. This point you must understand. Whenever time permits, you must visit such hospitals and know the details of the available facilities in such places, see the equipment available. All these things will go a long way in understanding the subject. I just want to briefly tell you about this COVID condition because it is one of the respiratory intensive care thing which is currently being debated and then discussed. COVID-19 can affect the upper respiratory tract, that is, it affects the sinuses, nose, and throat, and lower respiratory tract, the windpipe, and the lung. This is one of the main problems, and then which we are facing since the last 1.5 years. Here, you must understand that they are using the swab to collect the sample of secretions available in the sinuses, nose, and throat. So they put one swab in your nostrils and another one more swab in your throat to collect the samples from this for the RT-PCR test. I think you must have undergone. Most of you are familiar with this particular process. And you must have remembered this particular thing. And when you are here and then you went for these tests. To be very brief about what exactly is happening. And then the COVID-19 virus, that is SARS-CoV-2. And this is the viral particle, which is very minute in size, and then they get tagged on to receptor, that is angiotensin converting enzyme 2 receptors. So these things will get attached here, and then subsequently will be causing infection in the host cell. And then they cause pro-inflammatory cytokine profile is altered, and microangiopathic vasculopathy is affecting, and then specific antibody development occurs. So in short, the viral particle will get tagged on to the angiotensin converting enzyme receptor, and subsequently this will initiate the process, and then this results in pathophysiological changes occurring in the system. Actually, all these COVID patients are subjected to CT scan of the chest. Whereas you'll be taking the chest X-ray, and then besides the RT-PCR, you can also come to a diagnosis of the COVID infection in the patient by lung CT. So this is broadly divided into 5 grades. And this is CORAD 1, CORADs, that is COVID radiology, CORAD 1, 2, 3, 4, 5, and 6. Suppose if in the radiological examination, if one is identified as CORAD 5 or 6, it is deemed to be an infective condition. And then if it is 1, 2, 3, 4, it is less infective condition. So this, in addition, generally with RT-PCR tests, it's normally being done. 14% of the COVID-19 affects lungs. 14% of COVID-19 affects the lungs. And lungs are filled with fluid. Lungs will be filled with fluid. And this results in pneumonia, wherein the alveoli are filled with mucus, fluid, and other cells. So this is the point you must remember, because somebody asks you about the details, you must be in question to tell. 14% of COVID-19 affect the lungs. Lungs are filled with fluid and pneumonia, and then alveoli filled with mucus and fluid and other cells. This is necessitating the need for oxygen, wherein oxygen concentration in the blood falls, and that is the oxygen saturation in the blood falls, and subsequently you have to give oxygen to these patients. There is a new variant right now, at the current on date, the new variant of the severe acute respiratory syndrome of coronavirus 2, that is SARS-CoV-2, which is new Delta plus variant. So new plus designation is because it is not quite different enough from the original Delta variant to merit its own Greek alphabet letter. So technically it is the Delta virus, Delta variant, which is responsible for the present infection. And mostly you will be seeing all these things much more in Maharashtra and other parts of the country. Mostly we found more, about the 40 cases were found in India, and most of them are from Maharashtra state. Coming back to the pulmonary physiology or respiratory physiology, which the topics which will be covered to you in subsequent classes in physiology are about the muscles of respiration, that is muscles concerned with inspiration and expiration. And we'll also be learning about the elastic properties of the lung, that is pressure volume curves and lung compliance and tissue elastic recoil. And you'll also learn about the surface tension of a substance elaborated by type 2 pneumocytes in the alveoli of the lung. And the regional differences in ventilation, that is the ventilation definition, and differences in the ventilation vertically down the upright lung, and small air closure, etc. And you will also learn about the elastic properties of the chest wall. And knowledge of pulmonary physiology will be useful in branches like anesthesia. That is, a sound knowledge will be useful in these things. General medicine, pediatrics, cardiothoracic surgery, and pulmonary medicine. And principal functions of the respiratory system are mainly to ventilate the lungs and extract oxygen from the air and transfer it to the bloodstream, and extract carbon dioxide and water vapor, maintain acid-base balance. Just remember, why should you have the respiratory system? So the main purpose of the respiratory system is to ventilate the lung. Ventilation is different from oxygenation. Ventilate the lung, extract oxygen from the air and transfer it to the bloodstream, and excrete carbon dioxide and water vapor, and maintain the acid-base balance. Note down these things, and whenever in the Viva they ask you about the principal functions of the respiratory system, you should be in position to tell this. What is the composition of inspired air gas? That is, inspired air contains approximately 79% nitrogen and 20% oxygen and 0.04% carbon dioxide and water vapor traces. Whereas the expired air gas contains 79% nitrogen again and 16% oxygen and 4% carbon dioxide. So you just remember that inspired air doesn't contain much of carbon dioxide, whereas the expired air contains more of carbon dioxide, 4%, 0.04. You remember these things, 4, if you remember, 0.04% of carbon dioxide and 4% carbon dioxide in the expired air gas. And oxygen in the inspired air is about 20%, and here it is about 16%. And water vapor will be in traces in the expired air gas. Here you must understand, among the topics which will be covered in subsequent classes is that you have to understand the term homeostasis. And the homeostasis, the term homeostasis is the maintenance of the constancy of the internal environment. And you should also remember, hemostasis is different. That is arrest of blood bleeding. Arrest of blood leaking is the hemostasis. Homeostasis is maintenance of the constancy of the internal environment. And the other thing is about the energy release, that is the carbohydrates combined with oxygen combination, the oxidation process wherein the carbon dioxide and water molecules are formed. And what exactly you understand by the gills and the artificial gills, you will be learning later. And here you'll also learn about what are the changes that are occurring in the respiratory system of the newborn. And what are the functions of the respiratory system? These things you'll be learning. And what are the primary functions and what are the secondary functions of the lung? The basic living unit of the body is a cell. The basic fundamental unit is the cell. There are about 100 trillion cells in our body. Though it is the fundamental unit, there are about 100 trillion cells in our body. The cells are continuously bathed by the extracellular fluid, which is called the internal environment. So this point you clearly understand at this stage. The basic fundamental unit is the cell. there are millions of cells in our body, and the cell, outside the cell, you have the fluid. That is the extracellular fluid. A cell, extracellular fluid. And the entire thing together is called the internal environment. The constituents of this fluid are controlled, and then they maintain the normal range of these constituents. The constituents of this fluid, that is the extracellular fluid, are controlled and they maintain the normal range of their constituents. So they maintain the constancy of the carbon dioxide molecule, they maintain the constancy of oxygen molecule, they maintain the constancy of chloride ions, they maintain the constancy of bicarbonate ions. All these things are maintained constant. That maintenance of the constancy of the internal environment is the homeostasis. The individual cell is capable of living and performing the task as long as the extracellular fluid is constant. Individual cell is capable of living and performing these tasks as long as the extracellular fluid is constant. So extracellular fluid got to be constant to normally function. So this point you must understand. So this is often a short note question which is being asked in your examinations. Claude Bernard. So he is the great scientist who has the professor of physiology. The environment in which our cells exist is the internal environment. The term was first used by Claude Bernard. Note down this, Claude Bernard. And sometimes may be asked in the Viva, sometimes may be asked as a short note question. And this is about the internal environment. So Claude Bernard coined the term internal environment, provided stable media necessary for the normal functioning of the cells. So they should be, for example, in summer, you maintain the temperature of the room by using air conditioner. So in the same way, so they got to maintain the constancy of the internal environment for the effective functioning of the cell. The internal environment should contain the correct concentrations of oxygen, carbon dioxide, hydrogen ion, nutritional materials for normal functioning. So the internal environment should have all these things for the effective functioning and effective working of that particular cell, whether it is nerve cell, whether it is endocrine cell, or whatever it is. So this is another gentleman, Walter B. Cannon. Note down. He is an American physiologist, Walter B. Cannon. Sometimes his name will be mentioned in the short notes, and then you'll be asked to write about that. Maintenance of the constancy of the environment. There it is only the internal environment. So the term internal environment is coined by that gentleman. So he says you have to maintain the constancy of the internal environment. So this gentleman, the American physiologist, is Walter B. Cannon, who used the term homeostasis. Homeostasis is ensured by several systems of the body. For example, partial pressures of carbon dioxide and oxygen are regulated by the lungs. Kidneys and acid-base balance, everything is maintained by the respiratory system and kidney and various systems in the body. And this is called homeostasis. And here, sometimes you'll be confused with the term hemostasis. Note down. Homeostasis, and this also you note down. Hemostasis, that is arrest of the escape of blood. I would suggest you, this is not the part of the subject, to have a desktop in your house and then sit there with your notebook and keep the textbooks also available and note down the important things which we'll be discussing, so that You can go through whatever you did not understand at this point of time. Actually, this is the, we are talking about the alveolar capillary membrane. It is 0.5 microns in diameter. So this is the alveolus, this violet part is the alveolus, and this is the capillary part. So alveolar capillary membrane, through which the gases will be diffused. So you have the alveolus, and then you have the alveolar epithelium, and you have the interstitial space here, and then you have the capillary epithelium here up till this. And here the partial pressure of oxygen in the alveolus from the inspired air gas is 104 millimeters of mercury. These values you must remember, note down. 104 millimeters of mercury in the alveolus, the partial pressure of oxygen in the alveolus. And always it flows from a higher potential to a lower potential, higher gradient to a lower gradient. So 104 to where in the red blood corpuscle, this is the red blood corpuscle. The diameter of the red blood corpuscle is 7.2 microns in diameter. So from 104 millimeters, it will be diffusing through the alveolar capillary membrane into the red blood corpuscle, where it is only 40. Here in the red blood corpuscle, the carbon dioxide concentration is 45 millimeters of mercury, whereas that in the alveolus is 40 millimeters of mercury. Consequently, from a higher potential to the lower potential, it moves from the red blood corpuscle onto the alveolus. This point you must remember. This is a very fundamental and important diagram which you must practice, so that whenever any question is asked, try to reproduce this and then use color sketch pens wherever it's possible. And this is the alveolar basement membrane, and this is capillary basement membrane. This is the basement membrane. And this is the capillary endothelium, as I mentioned before. And this is the red blood corpuscle. The diameter of the red blood corpuscle is 7.2 microns in diameter. And this is the capillary. The keyboard panjayta. Alveolar capillary membrane is nearly about 0.005 millimeters in diameter, or it can be said 0.5 microns. Whereas the red blood corpuscle is 7.2 microns in diameter. You have to remember these values, which will be very useful in understanding. And this is about the fetal circulation. I think you must have learned in your cardiovascular system. Fetal circulation, whereas as a newborn is born, you have the placenta that is formed, and then there is a uterine area. Here it is called the blood barrier. Whereas the alveolar capillary membrane is 0.5, whereas the blood placental barrier or blood barrier is 3.5 here. So this value you must understand. This is much thicker when compared to the alveoli. So the blood from the mother's uterine wall is taken in, and then through it goes into the placenta. From the placenta, it passes through the our vessels and then goes to the fetal circulation. So in physiology, there are different barriers, note down, different barriers. That is blood placental barrier, blood aqueous barrier, blood testis barrier, blood testis barrier, blood aqueous barrier, and blood placental barrier. All these are the different barriers. There are a few more barriers which I will be communicating to you during the subsequent classes. So here, you always try to remember and then try to reproduce some diagrams. The uterine wall, this is made of smooth muscles, and then you have the placental septum, and you have the chorionic vessels here, and you have the umbilical vein. through which it goes into the fetal circulation. So ventilation, you have the trachea divides into two branches, and then this is the pulmonary airway tree, and then you have the bronchus, and then subsequently smaller bronchus, terminal bronchioles, respiratory bronchioles, and alveolar ducts, etc. So this is the division of the trachea into smaller units. Tracheal bronchiole, and then fundamental unit alveoli is attached to the terminal bronchioles or the respiratory bronchioles. The lungs are the organs of gas exchange between the blood and the external environment. So ventilation doesn't mean that it is respiration. Ventilation means you are moving a part of the organs of the body wherein the air is brought in contact with the lungs. The lungs are the organs of the gas exchange between the blood and the external environment. The process of moving air from the environment in and out of the lungs is known as ventilation. Sometimes in the Viva, they ask you, what is ventilation? So you can mention that from the external environment, as you are taking in air, which comes in contact with the alveoli or the fundamental units of the respiratory system, is called the ventilation. And diffusion is different. So diffusion is the next step involving is the diffusion of gases, that is oxygen and carbon dioxide, across the alveolar capillary membrane, that is pulmonary diffusion. That is from the alveolar space, the oxygen molecule is coming down, and then it is stacked down to the hemoglobin. And then hemoglobin, it releases oxygen. And then this oxygen is utilized for oxidation, and then water and carbon dioxide are formed subsequently, plus energy. So you have to understand the thing, that is, ventilation is different, diffusion is different, transport of gases is different. So transport of gases in the sense, the blood circulation transports the gases between the lungs and the tissue. So blood circulation transport gases between the lungs and the tissues. That is the transport of the gases. Sorry. The chairman garu maatadaru. So this is the alveoli, and this is the venous portion of the plexus. That is the blue colored one, and the pink and these things is the arteriolar red. And as you breathe in air, it comes into the alveolus, and air inflates the alveolus, and then air comes in contact with the alveolar capillary membrane. Alveolar capillary membrane. What happens here is, oxygen is at higher concentration here, that is 104, and in the venous blood it is 40. So it diffuses from here to the venous blood. And here the carbon dioxide concentration is at the higher potential, so it moves from the venous blood into the alveolus. So oxygen enters, carbon dioxide leaves the blood vessel. So all these places it will have the same. And this happens in the alveolar capillary membrane. So you must remember, transport means from the alveolus, it is moving into the circulation, and then from the venous end, it gets oxygenated and then it goes up. So roughly about 3/4 of a second, the red blood corpuscle moves from one alveoli to another alveoli. That is to say, that is in less than 3/4 of a second, the red blood corpuscle, which is roughly about 7.2 microns in diameter, will be moving or will be traversing 3 alveoli, wherein the oxygen molecule is stacked onto the hemoglobin. At this point, you must thoroughly understand. Less than 3/4 of a second, 3 alveoli, the oxygen molecule will be attached onto the red blood corpuscle. Internal respiration. What exactly? So far, we are talking about the atmosphere, and then this is coming to the coming in contact with the alveoli. And that particular thing is external respiration. Finally, the tissues utilize oxygen and produce carbon dioxide. The last step, which may be called as tissue respiration or internal respiration. What is happening within the tissues is the internal respiration. So far, till alveoli, it is the external respiration, then the oxygenation of the Hb molecule, and ultimately the oxygen is released, and that particular thing is the internal respiration. So you have the surrounding media. The oxygen is attached to the respiratory organs, and then you have the carbon dioxide which escapes from the respiratory organs into the alveoli. And this particular thing is called the external respiration. That is, oxygen is acquired. Oxygen is added to a bank balance. From the bank balance, it goes to the tissues wherein the HbO2 releases the oxygen. Oxygen combined here with the glucose molecule, C6H12O6, and then the combustion takes place, and then the oxidation takes place, and then carbon dioxide and water molecules are released, and then you have the energy release. So subsequently, as the oxygen molecules enter, the bicarbonate ions which accumulate consequently will be entering into the respiratory organs. And this is the internal respiration. The carbon dioxide diffuses from the bloodstream to the alveolus, and oxygen molecules diffuse from the alveolus to the bloodstream and attach to the red blood corpuscle. So try to practice these diagrams, which may be useful when you are writing the theory part or the practical part. So how exactly the respiration or respiration is happening in different lower animals? So you must also remember here that none of the systems do act separately. None of the systems do act separately. That is, the respiratory system keeps on taking air. The air comes in contact with the alveoli. Alveoli, then it releases the entry of transport of the gases, and then subsequently the oxygen molecules are released in the tissues, and then combustion taking place. All these things are simultaneously happening. At the same time, as these changes are happening in the lungs, there is simultaneously both the ventricles contract, and from the right ventricle goes to the pulmonary circulation. From the left ventricle, it goes to systemic circulation. All these things are happening simultaneously. So the lungs are functioning, the cardiovascular system is functioning, and then everything is filtered, and then the kidneys are functioning, and then you have the formation of water molecules, and these water molecules collect in the pelvis of the kidney, then traverses down to the ureter, and then gets collected in the urinary bladder. All these things do happen simultaneously. But we are dealing with each system separately only to facilitate the easy understanding of the subject. As a matter of fact, the physiology is more difficult and more tough because it's more principles involved in different functions. Whereas anatomy is just the structure which will be learning and then memorizing this. So here the concepts you have to understand. So normally the amoeba is the first unicellular organism. So you have the pseudopodia coming out, and then it takes the food molecules, and this is digested, and then unwanted food material is ejected out. So this goes only in the unicellular animal. You have the nucleus, and then you have the contractile vacuole. Similarly, oxygen diffuses from the environment and then enters through the cell membrane and then enters. So the respiration is done by the cell membrane. Whereas the human beings are multicellular. So you have the trillions of cells, and then how do you get in contact with that? For which you have developed the respiratory organs. You must also understand that some amount of breathing does take place in the human skin also, but very negligible. And when compared to that of the lungs, when compared to lungs, we are mainly the respiratory organ, some amount of respiration do take place in the human skin. Man's skin is only about 2 square meters area, whereas the total surface area of the lungs is roughly about 50 to 100 square meters area. So you have much enormous area wherein the oxygenation can take place. Whereas in lower animals like amphibian, frogs and other things. So cutaneous respiration is one of the major things. That is, the respiration does happen through the skin. And the vital respiratory organs are less. So these are the amphibious animals, and then that is those animals which are capable of living on the earth and also in water. So the cutaneous respiration mainly facilitates and are very helpful there for the amphibians. And you have to understand this. The fishes live in water, aquatic animals. And the gills are nothing but very complex outgrowths from the outer coverings. So you have the gills, and then these gills, they swallow water, and then from the water, they extract oxygen molecules, and then unwanted things are being thrown out through the gills. So the gills are the respiratory organs in the fishes. And you must also remember, there are the outgrowths from the anterior part of the intestines. They are outgrowths from the anterior part of the intestine. Even all the things in the embryological development, they only grow from the anterior part of the intestine. The gills are nothing but very complex outgrowths of the outer coverings. The gills and lungs are lungs of all invertebrates are modification of the anterior part of the intestines. So you must remember, most of the respiratory organs are developed from the anterior part of the intestines. When you learn about the embryology, you will have much more depth of knowledge. One of the greatest feature of the human beings, that is a female, is the creating another individual like it. So after God, it's only the female who is capable of developing another. So this is the mother's uterus, wherein it is seen a full grown baby. So, and this is the placenta. And then here you have blood placental barrier, the blood of the mother's uterine wall, and then the placenta. This is the barrier, which is 3.5 microns in diameter. So this part you must remember. So the fastest growing thing in the nature is the baby. Normally, it is for a full grown baby, it takes 9 months, and everything goes in the mother's uterus. And then at the full term, the mother, it is accommodated in the uterus, and then the uterine wall. And this is the mucus plug just before the delivery, knotty phase. That is, there are different stages of labor. That is, first stage, second stage, and then third stage. These details you'll be learning in the later part of your medical curriculum. And then it is effaced here. It is not effaced here. And you have the mucus plug, and this is the cervical cervix, and this is the vagina. This is the vagina. And this is the growth which you will be seeing. As a matter of fact, in the fetal circulation, as the baby is in the mother's womb, the baby does not breathe. All the oxygen requirements are met by the fetoplacental blood placental barrier. And then through the blood placental barrier, blood is supplied. Mother supplies the oxygen requirement of the baby. Mother supplies the oxygen requirement of the baby. So the lungs which grow from the anterior part of the intestines develop are non-functional in the fetus. But they just, you will be seeing only some moments of the lungs periodically in the ultrasound. If you do the ultrasound of the mother's womb, and then you will be able to see the contractions of the lungs, which are not functional. And then you must remember that is, as the baby is born in the first stage and second stage and birth, as the baby is moving from the birth canal, first the head comes out. As the head comes out, the attending gynecologist or the pediatrician available will be cleaning the nostrils in the beginning, so that any secretions which are there obstructing the nasal passages are clear. And subsequently, what happens is, as the baby is coming out from the birth canal, the thorax is squeezed so that the secretions which are there in the lungs, are pushed out because of the squeezing action of the birth canal as the baby is being born in the stages of labor. So subsequently, the secretions in the thorax and the body are being pushed out. So the intrapleural pressure during the first breath may fall, intrapleural pressure. Normally, the negative intrapleural pressure of nearly -2 to -4 is roughly mentioning is necessary for the expansion of the chest. So there is what is called the pressure dynamics, which are also applicable during the process of inspiration, which is responsible for the expansion of the lung. So this intrapleural pressure during the delivery and during the first phase of breath of the new baby may fall very high to -40 to -100 centimeters of water before the air enters into the lung. So these are the dynamic changes which are occurring in the cardiovascular and respiratory system in the newborn. So unless these changes occur, the baby will not be able to survive. So there are only two possibilities at this point of time, whether they live or whether they die, whether they achieve or whether they don't achieve. So this is very important. That is, as it is coming out, it is squeezing out of the birth canal during the contractions, and then squeezing out the secretions which are there, and then the attending gynecologist or the pediatrician will be cleaning the nasal passages, and the precise minus negativity in the intrapleural cavity during that particular stage is mainly responsible for the first breath. So during that, enormous amount of pressure is required to for the air entry into the lungs. Then only it will be getting inflated. All the alveoli open up. Till such time, it is only a mass. It is only a mass which is non-functional. So in the postmortem, sometimes in the forensic medicine, you have the situations wherein you try to understand whether the child is born stillborn or born alive and then killed. So this information will be very useful. So whenever you are studying medicine, you must also have a analytical approach and then a doubting the thing, why this is happening. Then only you'll be able to understand things in a much better way. As I told you, the first inspiration must pull 36 times more viscous fluid and then 1000 times denser secretion than air, so that the air enters in and then opens up. So it is something like a collapsed balloon, and then you blow air into that. So you have millions of alveoli which got to be opened up in the similar fashion. So that is happening in the first breath of the baby. And this is another picture where the blood placental barrier, the blood barrier, approximately 3.5 microns in diameter, whereas the alveolar capillary membrane, which we discussed earlier, is only about 0.5 microns. This is 7 times more. It takes more time. So you have the placenta, and then you have the umbilical vessel. And this is the placenta, which is there. In the 3rd stage of labor, the placenta is removed. And this is the placenta. Normally, what happens is, as the baby is born, the placenta is ligated. The umbilical cord is ligated. So that various things to happen. Suddenly, what is happening is the baby is receiving. The baby, once the umbilical cord is tied. So what is happening is the baby is no longer receiving blood from the placenta. So you have tied everything. And there is a fall in the oxygen, and then there is a rise in the carbon dioxide tension in the blood of the baby. And then at the same time, as the baby is coming to the new environment in which we are living, so suddenly it is bombarded with different stimuli. All these things are acting in a tandem manner, and then that is resulting in the dynamic rise in the carbon dioxide tension. And this rise in the carbon dioxide tension is stimulating the pneumotaxic center in the brain. So that is, the brain is developed, and then that particular thing initiates the respiration in the baby. So these are the dynamic things which occur in the baby as the baby is born. And this is the placenta, which is normally coming out, and then post-delivery. And then you must also know about the different structures of the placenta. You have cotyledons, and then blood sinusoids. All these things you'll be learning in anatomy. So this is the umbilical cord, and then it supplies the placenta, and then this is a uterine blood vessels, wherein it is thrown into sinusoids. And waste and carbon dioxide is delivered from the baby. Nutrients, hormones are delivered. And this is how the growth is seen in the mother in the cross-section. So you must here understand ductus arteriosus. You must have learned in your respiratory cardiovascular system about the ductus arteriosus. So the resultant, the details you must be familiar with. With the resultant increase in the blood flow from the left atrial pressure rises, and the flap-like foramen ovale quickly closes. This is helped by the fall of the right atrial pressure. As the umbilical flow ceases, the ductus arteriosus constricts a few minutes later in response to the direct action of the increase in the oxygen tension on the smooth muscle. So in short, what is happening is, so suddenly the right atrial pressure rises. As the right atrial pressure rises, and then from the atria, blood enters into the right ventricle. And then you have the septum primum and septum secundum, and then all these things are there. And then that approximation will be resulting in the closure of the foramen ovale. And you have noted, these two layers will close and then ultimately become the interventricular septum. So this point you must remember, and subsequently because of the oxygen, increased oxygen levels, the ductus arteriosus constricts and then closes. So there are different concepts mentioned for the closure of the ductus arteriosus. Because of the prostaglandins, prostaglandin E, prostaglandin F, and there's some unknown reasons. All these things are mainly responsible for the closure of the ductus arteriosus. In some abnormal conditions, you still have the ductus patent, and that abnormal condition is called patent ductus arteriosus. When you go to the clinical side, when you go to the cardiovascular system medicine posting, you'll be learning about these things. Some abnormalities in the pediatrics wherein sometimes the ductus arteriosus remains patent. That particular thing is called patent ductus arteriosus. So this is the foramen ovale, which I am telling. The left atrial pressure rises, and then subsequently, because of the rise in the pressure and because of the pushing, there is the closure of these two things, and then approximation, and subsequently it becomes the interatrial septum. So you must be familiar, you must understand some of the new terminologies and then note down all these things. Hypercapnia is deep breathing. Hypercapnia. Hyper means, always remember, the word can be divided with the prefix and suffix. So the prefix hyper means more. Whenever you come to the thing, hyper means more. Hypo means less. These are the two things which you should be familiar. And then everyone should buy a medical dictionary. The standard medical dictionary is Stedman's medical dictionary. So that gives a detailed information. Whenever you have any doubt about this terminology, you always refer the Stedman's medical dictionary. Dyspnea. So you must also know the spelling. You must also know how it is pronounced. Dyspnea is the rapid and shallow breathing of which the patient is conscious. So normally under quiet respiratory conditions, you are not conscious about your breathing. You are quietly sitting, quietly listening to my PowerPoint and online classes. So that is quiet breathing. Suppose if you are conscious of your breathing act, it is called dyspnea. It is a shallow breathing. Or sometimes it is called undue consciousness of the breathing act, dyspnea. Apnea is the period of respiratory arrest. Apnea means absence of breathing. That is, can be voluntary, can be because of pathological conditions. So you must note down all these things which are mentioned in the pink color. That is hypercapnia, dyspnea, apnea. All these things may be asked in the viva or may be asked in the short note question. Just like a neuron is a fundamental unit, a nephron is a fundamental unit of the kidney, a neuron, fundamental unit of the thing. So sinus is a fundamental, alveoli is the fundamental unit. Sinus is a portion of the lung supplied by the terminal bronchiole. So alveoli, nephron, neuron, all these things are the fundamental unit. It's nothing but the modification of a cell. The cell is modified, wherein it results in the neuronal cell or endocrine cell or the respiratory cell. Sinus is a portion of the lung supplied by the terminal bronchiole, of which there are nearly about 64,000. It is called as sinus. So a terminal bronchiole is a number of things. Let us say, if you can compare number of small balloons, which are coming from a single trunk. So that particular thing is called the sinus, which roughly about 64,000 will be there, and is the functional unit of the lung, wherein the gas exchange is taking place, and each consists of branching of the respiratory bronchioles and leading to cluster of alveoli. So this is the diagrammatic representation of the useless. This will be distracting. Bronchiole, this is a pictorial thing of the SNS. So this is about the SNS. Note down SNS and other details. And this is the pictorial thing, wherein you have the bronchiole and then this divides, and then you have the number of things in the, and you have the capillary beds around these alveoli. As I said, roughly in 3/4 of a second, it traverses through 3 alveoli. The red blood corpuscle, which is 7.2 microns in diameter, will be traversing through 3 different alveoli to get oxygenated in less than 3/4 of a second. So this point, the dynamics of the movements of the blood flow in the pulmonary capillary. And then remember this diagram. And you have the red blood corpuscle, the cross section of the lung. And then you have the alveoli, something like a bunch of grapes. And this is about the structure of the trachea, and then what are the different things in the trachea you must know. And up till this, the thyroid cartilage and cricoid cartilage, this area, the larynx. So larynx is different, trachea is different. Get familiar with these things, read about these things, and draw a diagram, and then keep it in this. Thyroid cartilage, and then cricoid cartilage, and cricothyroid membrane. And then you have the trachea, tracheal rings, and the annular ligament, tracheal cartilage, and then right bronchus and the left bronchus. All these things are happening at the angle of Lewis. Angle of Lewis is between the manubrium sterni and the body of the sternum, wherein it's slightly angulated, and wherein you find the bifurcation of the trachea occurring at that level. And this is the cross-section of the trachea, as is shown in the glass picture here. So that is, you have the trachealis muscle, and then you behind that you find the esophagus, and then you have the lumen, and then the lumen of the trachea, and you find I think once you come to the anatomy thing, you have the histology, wherein you find all these type of histological pictures, wherein the pseudo-stratified ciliated columnar epithelium. So here in the respiratory passages, pseudo-stratified ciliated columnar epithelium, whereas on the skin you find the stratified epithelium, and then ciliated means where the movements are required. So once you study the different thing and then analyze critically about the terminology, you'll be able to understand that with much ease. And this is the C-shaped cartilage. And the alveoli and the gaseous exchange is taking place here, and then you have the blood circulation, and then this is the alveolus. This is again another picture of alveolus, and then the erythrocyte of the red blood corpuscle will be traversing here. And you have the alveoli and then the bronchioles. So there are, in lungs, the lung per se, you have 2 different types of cells. So there are further division of all these cells. I think at this point of time, you can understand. When you are talking about the lungs, when you are talking about the fundamental cell, it is the pneumocyte. So in neurons, and here it is the pneumocyte. There are 2 types of pneumocyte, that is type 1 pneumocyte and type 2 pneumocytes. In addition, in the lung, there are macrophages, the plasma cells, the lymphocytes, the mast cells in the lungs. Again, there are 2 different types of pneumocytes, that is lung cells. That is type 1 pneumocyte, type 2 pneumocyte. Remember this, sometimes in the examination, they just give you type 2 pneumocytes, wherein you have to mention a lot of details mentioned here. In addition, there are macrophages. In addition to these type 1 and type 2 pneumocytes, you also find macrophages, the plasma cells, the lymphocytes, and the mast cells in the lungs. So macrophages, plasma cells, lymphocytes, mast cells in the lungs. Type 2 pneumocytes are of interest for us. Type 2 pneumocytes are thick with numerous cytoplasmic inclusions and secretes surfactant. So in the normal histological picture, whenever you find more numerous cytoplasmic inclusion, that means This cell is a secretory cell. So the other cells are normal. So normally a cell which secretes something will have more cytoplasmic inclusion in the histological picture. And these produce what is called the surfactant. Remember the word surfactant? The surfactant is commonly asked question in the examinations in the viva or in the short note question, where you have the details of these things but I mentioned that it is elaborated by type 2 pneumocytes in the lungs. And this particular surfactant is mainly responsible for maintenance of the pressure dynamics. And here you must also remember Laplace law. Note down. This is the Laplace law. So Laplace law, the smaller balloons with greater pressure empties into the larger balloons. Smaller balloons with greater pressure empties into the larger. These are the dynamics. And then this is governed by what is called the Laplace law. The pressure in the lungs is equal to twice the surface tension divided by the radius. Pressure in the lungs is equal to 2 times the surface tension divided by radius. Surface tension is generally in physics, you must remember, intermolecular attraction. The surface tension is nothing but intermolecular attraction, which you will be finding in the primary factor and causing elastic recoil of the lungs. Surfactant made by the type 2 cells reduces surface tension and stabilizes the lung alveoli. So this surfactant is mainly responsible for stabilizing the lung alveoli. Alveoli remain in a patent condition, remain in a opened situation is mainly because it is lined by the surfactant. Had there not been a surfactant, it would have collapsed. Had there not been a surfactant elaborated by type 2 cells, the alveoli would have collapsed. What is the chemical nature of the surfactant? Surfactant contains dipalmityl phosphatidylcholine. So sometimes they just give you, instead of mentioning surfactant, they just mention dipalmityl phosphatidylcholine. So you must tell about this chemical nature of the surfactant substance. A detergent synthesized by the type 2 pneumocytes. Together they cause the hysteresis seen in compliance curves. Note down, hysteresis is another important thing which you must understand. That is, the inspiration curve is different from the expiratory curve. This particular deviation is the phenomena is called hysteresis. So mostly the inspiration is an active phenomena, whereas the expiration is a passive one. I told you that inspiration is 3 seconds, whereas the expiration is only for 1 second. Impedance of the blood flow also you must be able to understand. So you must hear important things is surfactant and dipalmityl phosphatidylcholine, Laplace equation, and the pressure in the lungs, that is twice the surface tension by radius. So this is the normal airway. You have the type 2. Sometimes the type 2 pneumocytes are called Clara cells. Note down. Type 2 pneumocytes are sometimes also called Clara cells. C-L-A-R-A, Clara cells. Sometimes they just give you Clara cell. What exactly you have to mention is about the type 2 pneumocytes and where exactly it is situated, what exactly it elaborates on, what exactly it functions. So this is the air, and then mostly all these noxious things and bigger particles are filtered in the respiratory passage. The other cells which you are finding in the alveoli, this is the pneumocytes, alveolar type 2 pneumocytes, and then type 1 pneumocyte here. And then you have the macrophages, and then you have the plasma cells. Everything will be there in this particular picture. So that's the reason why I said you try to practice the diagram so that you remember these things in much better way and keep doing it whenever you find time. This is about the internal respiration and then external respiration. That is normally what happens is blue colored one depicts the deoxygenated blood and the red colored one depicts the oxygenated one. So normally happens is you have the superior vena cava. You just remember, concentrate here. You have the superior vena cava, the blood from the upper portion of the body is collected. And you have the inferior vena cava, that is blood from the lower portion of the body is collected, and then that goes to the right atrium. As the right atrium gets filled with blood, it enters into the right ventricle. From the right ventricle, as the ventricle contracts, the blood enters through the pulmonary veins into the lungs. There you have the pulmonary capillaries, and then you have the trachea, bronchi, bronchioles, and then the alveolus, and then ultimately reaches the terminal bronchiole, and then you have the sinus, and then you come to the alveolar capillary membrane where the oxygenation is taking place. And then it is moving from the external that is atmosphere down into the blood. In the blood, it is stagnant to the hemoglobin molecules. And then they transit through 3 different alveoli, the blood corpuscles, 3 different alveoli, and then catch the oxygen and getting saturated. And then all these things again, after oxygenation or will be entering into the left atrium, entering into the left atrium, and then from the left atrium, it enters into the left ventricle. In the left ventricle, as the ventricle contracts, it's pumped from the aorta into systemic circulation and then goes to different parts. So as it is traversing from different parts, the oxygen is released into the tissues, and then in the tissues, it is combining with C6H12O6, and then it results in the combination in the oxidation, and then releases carbon dioxide and water molecules plus energy. So this energy is used for cell building. This energy is used for biological activities which are occurring in the cell. Cell energy is required for the synthesis of hormones. Energy is required for the activation of the neuronal cells. All these things, ATP molecules are released, and these is required throughout the body for energy supply. So this is in short about the external inspiration, and this is the external inspiration, and then ultimately what is happening in the tissues is the internal inspiration. Again, the explanation of the tracts. There are different sinuses: nasal cavity, mouth, pharynx, and epiglottis, alveoli, the lungs. As I said, this is the horizontal fissure, right lung, and then this is the oblique fissure. This is the oblique fissure in the left lung, 10 lobes, and then the 8 lobes. All these things you must remember. Again, another elaboration of the respiratory tract. So this is a picture which I was continuously referring to, so that if you can practice all these things, the right lung has 3 lobes and 10 bronchopulmonary segments. And then the left lung has 2 lobes and 8 bronchopulmonary segments. If you do these things about 3-4 times in your rough notebook, you remember that it is horizontal fissure and then the oblique fissure. All these things you can map. So in normal learning, what is happening is you have to read, and then you have to reinforce that particular reading. Then only it will get imprinted in the brain. So just one reading or one listening or one visual glance will not be sufficient to remember things. So you have the trachea and then different parts. And then you have the pleura. Pleura is nothing but the covering of the lung. So something like a polythene sheet which covers the lung. So that which covers the lung is the visceral pleura, and that which covers the wall is the parietal pleura. So in between you find space that is called the pleural space or pleural cavity. So normally what happens is in abnormal conditions, water gets collected in this pleural cavity, and that is called the pleural effusion. Normally, to get back to the present COVID scenario, it is the lung parenchyma, which is normally involved, and then the alveoli are filled with fluid here because of the COVID virus. And then this fluid impedes the normal oxygenation, and then that results in the fall of the oxygen tension. So this is again the pons, and then this portion, lower portion, the medulla. And you have what is called some respiratory centers. The respiratory centers, in the sense, they are the centers which mainly control the respiration. So you have the pneumotaxic center, and then what is depicted in the red color is the pneumotaxic center, and then this is the apnostic center. Normally, remember these things, note down this pneumotaxic center and then the apnostic center. So normally the apnostic center sends signals to the dorsal respiratory group. Pneumotaxic apnostic center sends signals to the dorsal respiratory group, that is the inspiratory center. So when the inspiratory center in the brain is stimulated, it sends signals to the intercostal muscles, external intercostal muscles. So external intercostal muscles are the inspiratory muscles. And these inspiratory muscles will be acting, and they result in expansion of the chest wall and subsequently sucking air from the atmosphere into the lungs. So that is what is happening. So in short, remember, the details you will be learning in the late portion, but still at this point of time, if you should understand. So that is apnostic center. Apnostic center stimulates the inspiratory center. The inspiratory center contains the nucleus or the nerve cells which are situated in the dorsal respiratory group, and that in turn will be stimulating the muscles of inspiration. The muscles of the inspiration are the external intercostal muscles. So the other day, Dr. Chari was telling about the intercostal muscles, the attachment, and then these things. So it is these muscles which are getting stimulated. So normally this, once this is achieved, this is the, will be stimulating the expiratory center. Expiratory center is the ventral respiratory group. That which is located dorsally is the dorsal respiratory group. That which is located ventrally is the ventral respiratory. Ventral respiratory group will be stimulating, and then will be stimulating the muscles of expiration. But most of the activity of expiration is passive. Here you must recollect, that is, inspiration is 3 seconds. Expiration is 1 second. Inspiration is longer than expiration. Expiration is passive phenomena. Inspiration is an active phenomenon involving external intercostal muscles, where there is eversion of the ribs and the movement of the diaphragm moves down. And then in addition to these things, the scalene muscles and other muscles will be also be active. Here you must understand that once the inspiration has reached a stage, the pneumotaxic center, which is inhibitory in nature, will be stimulating the apnostic center to stop the work. That is, in other words, it stops or it gives a negative feedback. The details of the negative feedback you will learn once the stimulation and other things here are being taught to you. But still you remember, pneumotaxic center will inhibit the apnostic center. So apnostic center no longer sends signals to the dorsal respiratory group, and dorsal respiratory group no longer sends signals to the external intercostal muscle. Hence it stops. Then the expiratory center is stimulated, and most of the expiration is passive. So you have the inspiratory center, which is mainly responsible for the inspiratory act. And in addition to the external intercostal muscles, you also have the diaphragm and then the scalene muscles, and then the manubrium sternum moves forwards. All these things will be giving a three-dimensional picture, which is enhanced. This is in detail. Wherever possible, try to get good pictures from the textbooks. And if the textbooks give very good picture, you take it. Get a Xerox copy and paste it in your notebook. So you can read your notes much faster than the textbook. So all these things you must practice from now onwards. As I said, agnostic center, and then that results in the stimulation of the dorsal respiratory group, and that is the inspiratory center. And then this in turn is inhibited by the vagus and the glossopharyngeal nerve. There is inhibition from the vagus nerve and then the glossopharyngeal nerve also. And the ventral respiratory group is the expiratory one. And then you have the mostly this a passive phenomena, the elastic recoil of the lung. So there are different paranasal sinuses. Sometimes in anatomy, they just give you paranasal sinuses. You have to mention about the frontal. That is, you have the paranasal sinuses, and then the ethmoidal sinuses, and then the sphenoidal sinuses, and then the maxillary sinuses. So these are the paranasal sinuses, which are spaces within the skull bone. So that's the reason why I said try to collect a skeleton. Try to procure skeleton from whichever source is available, and then try to read individual bones. In this COVID scenario, in this particular online phenomenon, so that you will complete that particular portion. So a thorough understanding of the skull, thorough understanding of the different portions of the skull is necessary for understanding anatomical things. So these are all the paranasal sinuses, that is frontal, ethmoidal, sphenoidal, and then the maxillary sinuses. So this is again the thyroid and other things. And this is the cross-section of the head. And this is the base of the tongue, epiglottis, and all these things you'll be learning. And they're learning endoscopic view. Note on these things. Mechanics of breathing. Muscles of respiration. That is, you have in the restful conditions, inspiratory muscles are diaphragm and external intercostal. Mainly external intercostal muscles. In addition, the diaphragm moves down and pushes all the viscera, so that the space in the above downward space is increased. In forced inspiration, for example, you are subjecting yourself to a fast running to your top floor of the lecture hall, and you are forced to take more than 250 ml of air, and wherein the accessory muscles of the neck are coming into action. Thorax and accessory muscles of the neck, thorax, and then the abdominal muscles also come into play. And expiration is mostly passive because of the passive recoil. In addition, when it is necessary to push out the air within the lungs, it is the internal intercostal muscles, then the neck, and then the abdomen. You have the pressure volume curves described, and then that is the compliance of the lung, which you'll be learning later. Factors involving the lung compliance is surfactant. And here, I just wanted to tell about the inhalation. So it is the scalene muscles elevate first to ribs, and then the inferior part of the sternum moves anteriorly, and external intercostal, and these are the muscles of the expiration. Transversus thoracis and depresses the lips. And these are the muscles of the back. Again, another picture so that you understand in a better way to recollect. This is about the artificial respiration. Just remember rocking method. Details you will learn later. This is again artificial respiration, mouth-to-mouth breathing. So all these things are the mannequins. That is, you have the models, and you can practice on these things. So there will be what is called the skills lab in each medical college, and then you can go to the skills lab and learn all these procedures whenever you find time, if you are inquisitive in learning. And this about the pulmonary circuit again, only another picture so that something will reinforce your memory. That is right atrium, blood moves from the right atrium to the right ventricle, from the right ventricle it goes to the pulmonary circulation, pulmonary artery, and the vena cava, right ventricle, and after the oxygenation, it comes to the left atrium and then left ventricle and then into systemic circulation. This is the histological picture. So normally in the histology, they are replacing respiratory bronchiole. And this is how typically it is displayed. In the undergraduate and postgraduate subjects, it will be placed and then you will be learning about these things. This is about compliance, early at this point of time, uncompliant. This is the surfactant diapalmityl phosphatidylcholine actual structure, which reduces surface tension, wherein the electrical attraction or the water molecules, which is normally available, is reduced by the presence of these chemical substances. This composition of the diapalmityl phosphatidylcholine alveoli surfactant. That is, had there not been a surfactant, it would have just collapsed. Because of surfactant, it is remaining patent. All these things we'll learn later. Okay, take care and then read well. God bless you.